Up to now, the only way to study the disease has been either look at brains from people who've died of the disease, which is useful, but you're not seeing the disease, you're seeing the endpoint. So we needed a way to look in human nerve cells in real time as the disease progresses.
So they did something amazing. Building on pioneering research done here 50 years ago, they took stem cells, the sort of cell that can form any other type of cell, and made them develop into brain cells.
They're all talking to one another, and they talk to one another in the same way they do in your brain or my brain. So these are live.
I know you do this every day, right?
Yeah.
So you're used to this, but it's mind-blowing what's going on in this laboratory. The fact that you can do this is humongous, isn't it?
Yeah, it's great fun. I mean, this is two-dimensional. No, it is. But also, so the other thing we're working on is, the brain is three-dimensional. So we're also busy trying to make this in 3 dimensions as well to get all the architecture. So making mini brains.
I mean, that's a little bit crazy. Are you telling me that in a Petri dish you can actually have a 3D brain?
Yeah.
Rick uses stem cells from someone who is genetically almost certain to get Alzheimer's, and he can effectively grow the disease in his lab.
If you look at the brain of somebody with Alzheimer's disease and you cut a little slice, you can find 2 bits of pathology, and they're called plaques and tangles.
For the first time, Rick can now watch them as they form. Plaques are made up of tiny fragments of protein, which stop the nerve cells communicating with each other, causing the symptoms of dementia.
This is plaque formation that is characteristic of Alzheimer's?
Exactly. So this is completely abnormal. So these are very sick neurons.
Tangles are involved in the spread of the disease. They're made up of a different protein present inside the brain cells.
In the disease, it actually just starts accumulating and forming these big, long tangles, which essentially strangle the neuron. But the other thing it does is we all now think it spreads from neuron to neuron, making the other neurons sick.
In another lab, Rick can watch the tangle protein passing on the disease.
It's not like an infection where a virus or a bacterium. This is a protein doing this, the simplest thing you could possibly do.
This is your body turning on itself. And why is it important to understand this?
Because we think this is a fundamental mechanism how the disease spreads through your brain.
And it's only because they can grow living brain cells that they've been able to see the disease spreading. This means Rick and his team can, for the first time, test a whole multitude of different drugs on live human brain cells and watch the effects on a cellular level. At the moment, over 1,000 drugs are being investigated.
How long before you're able to say to the pharmaceutical industry, this is one that's looking exciting, start making it?
And we're very close. We're now in the process of working out which 1 or 2 to really put our money on. That's within this year, effectively.
Within this year?
Yeah.
That's exciting stuff.
A life-changing treatment for Alzheimer's is now closer than ever.
You know, this is a scientific process at its best. Groundbreaking research that was carried out 50 years ago has now inspired others to apply that knowledge to find a cure for a debilitating disease. And it's a very exciting time for Alzheimer's research because this breakthrough, allowing us to understand the disease at its early stages using live human nerve cells, is completely transforming our understanding of how to find a cure.